white-fronted geese. So that involved a lot of field ecology, and I will credit the individuals that were involved at various times. So the work really looked at surveying geese, eyeballing, not using the high-tech tags, but with Gareth Thomas very often working hard with a high-power scope, watching the birds at different periods. And I'll talk about that in a little bit more detail, because that was key, identifying where the birds were located in each of these sites, to then following up with some detailed field survey with the plant ecologists and the nighttime poop collectors and so on involved. You'll hear more about that shortly. So it's quite complex in that from the field work I've just described very briefly, and I won't dwell on this diagram very much, but other than to say that a whole crescendo of other work took place. So from the collection of the fecal material from the geese, we used the DNA metabarcoding technique to identify the plant material. So Gemma Beattie and more recently Mark Scott was involved in the DNA extractions, PCRs, and then Andrew Detheridge has been critical recently dealing with the sequencing and interpreting the data. So I'm really pleased I can report some of the outcomes of that work shortly. And then there's a team which Holly Sayer's been working hard in recent months, but before her, Rod Kemp, very much doing a lot of the field collection and surveying work, but there's a much wider team, including Mark Scott, I mentioned earlier, helping with the processing of the plant material collected in the field, and leading up to Sue Lister and her team doing biochemical analysis, so that we had the dual outcome then of identifying which plants in the locations the geese were seen to forage are they actually consuming while they're grazing, and what is the nutritional value, of those plants to those geese during the winter months. So those are the areas we'll come into really. But work package 4 covers the tracking and the other aspects that you've heard about already from Catherine, and the results of those will come through in work package 3 and the presentation with the team involved in that later. So here's the spectacle of the Dovey estuary. We heard, why it's easy to see why the curlew love staying out on the estuary and not coming in much. I mean, the most they do is come in to the kind of shelf of the salt marsh when the tide is higher, but they occupy these vast sort of tidal sands and muds most of the time. But the geese are doing something different. They are overnighting on the saltings, the salt marsh, a little bit further up into the estuary, generally speaking. But they are using the fields you can just about see on the inland side of the railway line, mainly on the RSPB Unners Reserve, but also some other land a little bit to the west as well. And so that was the focus for that site. The character of the Kevney Valley on Anglesey was very different. So interestingly, most of the time we collected material and located the geese in that part of Anglesey, they were on flooded pastures. Sometimes they were on the RSPB Mathrive Reserve, but generally they were on quite intensively managed ryegrass of dairy farms, which happened to be seasonally flooded in the winter. They seemed to like the combination of the highly sort of edible leaf of the ryegrass, but in the context of waterlogged fields. And then this is the outlook from the observation tower at the sort of wildfowl reserve on the North Slob. And this is a very different situation because the two sites I've just shown you had between, say, 16 and 22 individual geese in a small self-contained flock that tended to stay as a group at wherever they went. But here we had up to 7,000 Greenland White fronted geese to observe. So it was more a point of looking at to sample areas where the geese were focusing their attention for grazing away from other species for reasons I'll come on to in a moment. So that's where we collected a lot of material. So Gareth Thomas and Tony Cross and myself have been over at various times to carry out the work there, with the tremendous cooperation of Dominic Berridge, who I would say needs particular credit, because he tended to do his day job, hang around, and then join us in the evenings and help with the sampling as well. So he had some very long days when we visited the site. So to give an idea then of the general strategy, Gareth Thomas would be spending the whole day of one of our target days. So we were aiming for an early winter, middle winter, late winter sampling period. And within each of those three, there were three separate targeted survey days. And so the idea was, we needed good visibility on those days. So it had to be reasonably clear weather. So we'd get the okay early in the morning. We're going to go for today. Gareth would generate maps and locate where the geese were with the telescope from a vantage point above Ferney's Egloisach, which is a little village near the RSPB visitor centre. And from there, he would be able to sort of get sort of 15-minute, half-hourly records of the goose locations. I would get a call sort of mid-afternoon where we would meet based on where the geese had been grazing. And with the combination of Gareth being willing to work a very long day and join me and walk out to the site and the maps, we would go out. And of course, we were looking for the goose poops. So what we had was a sort of, if I can get the mouse on screen, we had, roughly speaking, a map where we had lots of points where they'd landed from their overnight roost into a grazing field. And generally they wander along a headland or along a sort of area of grassland that they're feeding. And it is a fairly continuous line. And then at some point in the day, they depart back, usually dusk, depart back to the saltings where they roost overnight. Sometimes they might fly from here out to the saltings during the day, perhaps for a drink or a wash, and come back. And so there was that side of things as well. But we would target this main area and try to sample across that route. So what we're looking for in the dark with our head torch are the patches of lawn areas that Gareth, and it's very much relied not just on the map, but his intuition, because if you're looking down a telescope, you're only seeing everything in one perspective. And of course, the depth of field is quite difficult to judge. So he had sketches of where there was standing rush or fence lines and things. And so we would have to quarter to find these locations. But it meant that when we'd found the droppings, which are distinctly smaller than those of Canada geese, and we confirmed this with the DNA analysis later that we did collect the correct species poops. It also meant we had grid references we could record of their feeding locations on those days. The poops would relate to what they'd been eating perhaps 3 or 4 hours earlier. They have quite a rapid digestive sort of process, so the food's going through their tract quite rapidly, but of course it's relating to the plants they were eating a little bit earlier. But it also, where we picked them up, was absolute definite evidence of where they'd been feeding. And we collected, for every 4 poops in a cluster, we collected a bag full of the lawn material, which was for the vegetation analysis later as well. So that's the source material. And then Gemma Beattie with Mark Scott and Andrew Detheridge in particular, then selected out, and my understanding is they subsampled about 184 samples from our list, which I should have tallied, but we sort of collected multiples of that, several hundred poops over the season from the 3 sites. So they randomly selected for the 3 study sites for the early, middle, late winter, and for different locations that appeared in the data as well. And so the process involved using the QIAGEN TAC polymerase for the polymerase chain reaction. A lot of us know about that term now because of COVID and it being used in diagnostics, so it's actually much more familiar, but it basically amplifies a specific region of DNA, and then the sequence of the bases in that region are used to identify the plant species from a Welsh plant database that was generated by the National Botanic Gardens, Wales. And so That process, the actual process of sequencing, was carried out through contract with Cardiff University, and we used Illumina sequencing, where the bases are basically tagged with a fluorescent dye, and that just allows for extremely rapid reading of those individual bases. So some results. So from the 3 sites, the Dovey left, Wexford centre, Kevney right, and the Kevney is a combination of, it's a misnomer, I should say, on this morn, I guess, because it's some Kevney Valley, some in areas that were grazed around Llanello in the north of the island. So we've represented the main foraging areas. And these are generally typical grassland, meadow grassland species. So you've got at the top here, Lolium is the common ryegrass. So a lot of these pastures were probably reseeded 10 or more years ago, and they've slightly naturalized with native plant species, mainly grasses. And in there occasionally, Puxinalia is salt marsh grass. So this shows that the geese have been feeding out on the salt marsh, but the incidence of that tends to be later in the season. So we've picked that up in the late winter period. They're very much picking away at the range of species that are on these fields that, so in the Kevney Valley, those sort of flooded ryegrass fields. In the Dovey, the fields immediately adjacent to the railway line, but inland, so they're definitely terrestrial. Over at Wexford, again, they're foraging around stubble fields and grasslands in the sort of character that was depicted in the photograph that I showed you. So the percentage values are really just the occurrences of the genetic material, really, in the sequence analysis, and there's a lot more work to be done with this. But Andrew Delridge kindly just put together some very quick comparisons. So the dot diagrams at the top, the biplots, are just to illustrate really where there's differentiation of the individual sequenced data. So at the top, very left, is just 2 winters of data. So last winter and the previous winter, showing a high degree of overlap, really, which is something that we would hope to see, that there's some consistency of their dietary choice year to year, so it's predictable, basically. We can draw some conclusions. The middle one is looking at the different times, and there's a bit of separation there. So there is evidence of shifting in their dietary choice from early, middle to late winter. And as I said, one of the most standout differences, we haven't done the detailed analysis yet, but one of the most standout ones is that appearance of salt marsh species in the diet towards the end, which is consistent with things, with comments and observations from local bird experts like Dave Anning. I remember a discussion on this subject. And then at the end there, we've got the 3 different sites. So not surprisingly, the separation there, because you've got different botanical compositions, depending on whether it's more intensively agriculturally managed, like in the Kevney Valley, or whether we're talking about the sites in Ireland or in and around the south of the Dovey. So a little bit on the biochemistry now. So we have here the team, Sue Lister, who led this piece of work. So there are lots of us involved in sort of collecting and processing the work. So Holly's been working very hard, particularly sort of towards, I think it was the end of last winter, but full-time sort of devoted to this job through this winter sampling period as well, taking over from Rod Kemp, who left us in September. And but others too. So some are involved in more the lab and the processing, and others are involved in the biochemistry outside the field work. And the focus was really to process plant material, one from those collections in the grazing lawns that I described when we were collecting the poops. So that's Tony, Gareth and myself really primarily did that work. A bit of help from Rod, I think, a couple of winters ago. And then the other material is, Holly at the moment is targeting particular plants, because obviously if we just collect the material in the grazing lawn, it's a blend of different pasture or meadow species. So that gives us an idea of what the geese are getting from their intake from grazing. But we also need to know what the individual plant species contribute to the nutrition of the geese. So that's basically the different components that were measured from those 2 different sources of plants those collected from the grazing lawns, and the targeted plant species that we know feature in the diet of the geese. Primarily, that was gained from reading the literature before the study, and this winter we can target species that have come up from the results of the DNA metabarcoding. So there's not much difference between sites and over the 2 winters that I represent in these data. So you've got, it's basically last winter and the previous winter's data. We're still processing this winter. So you can see for nitrogen, there's some variation, but it's not really very significant, the differences overall. And there's very little differentiation in the gross energy, which sort of shows that the geese are selecting areas really that are benefiting them. You know, they're basically picking places that provide a standard sort of energy component in those grazing lawns. But what is interesting, so that's sort of just doing the overview look of site and year. But for the nitrogen, there's very clearly, I hope you can make out that there's a decline. So this is from meteorological winter day, is basically 1st of November. So it's basically just showing the nutritional value of that same grazing lawn material as you proceed through the winter. That shows quite a marked decline, implying that the birds will need to feed on proportionally more material to gain the same nitrogen, which is amino acid and protein, really important for muscle, obviously, and general growth. So that was significant for the nitrogen, but not significant for gross energy. That's overall energy content of the leaf material that they were consuming stayed fairly steady right through the winter. And then looking at different species, there is quite a substantial difference. So ironically, with the vegetation such as the common unsown lowly and perennial ryegrass, that does actually represent a higher value for nitrogen. So, you know, it seems a bit galling to admit that it's not the semi-natural species or native species, but it is the ryegrass, and Poa annua is early meadowgrass, and that also appears to be very important in terms of having overall a higher mean nitrogen as a proportion of the wet weight of the material. And then if I go down to gross energy, that doesn't vary greatly. You can see how quite similar it is per unit of plant material per kilogram. It's roughly speaking the same amount. But this one here is the kind of water-soluble carbohydrates. So this is your sugars and easily accessible energy in the material. That does vary. So you can see these are bents and fescues. That's the salt marsh sort of species. And then we've got, or aquatic vegetation, then we've got things like Yorkshire fog and ryegrass, early meadow, and that's the salt marsh grass itself. This is sweet floating grass, which we discovered they've been feeding on, and it's in the sort of drain margins and ponds. So that's actually come up as important from the genetic work, because when we surveyed the dry land. We didn't survey in the water areas. So just an overview, really. The top right, I'm not going to dwell on it because of time, but that's actually the less digestible. So the high columns are less desirable plants because